If you were to zoom inside a living cell, beneath the membrane and deep in its internal architecture, you'd find an extraordinary network of protein fibers working constantly to shape, move, and reorganize the cell. Among these fibers, one stands out for its speed, flexibility, and sheer versatility: actin. Actin filaments are the engines of motions inside nearly every eukaryotic cell. They form a dense glowing mesh just beneath the cell membrane, a powerful framework that drives crawling, stretching, division, and changes in cell shape. They're also intimately connected to proteins like myosin, forming miniature contractile systems that behave like microscopic muscles. Today we're going to explore how actin filaments grow, how they move the cell, how they help divide it in two, and why they're essential for everything from wound healing to immune defense. Actin filaments, also known as microfilaments, are the thinnest of cytoskeletal fibers, but don't let their size fool you. They're incredibly strong and incredibly dynamic. Each actin filament is made of tiny protein subunits called G-actin. When these subunits join together, they form long chains known as F-actin, or filamentous actin. These filaments assemble just beneath the plasma membrane, creating a tight, organized mesh that gives the cell its shape, tension, and flexibility. Actin filaments constantly assemble and disassemble, sometimes in a matter of seconds. This rapid remodeling allows a cell to change shape quickly, extending outward, retracting inward, tightening its grip, or loosening it. Actin is not a passive structure, it's an active force, a dynamic framework that lets cells respond instantly to their environment. To understand the power of actin, we need to zoom in even further, down to the molecular level. Each G-actin monomer carries a molecule of ATP. ATP isn't just fuel, it acts as a biochemical signal that tells actin when to assemble into filaments. When many G-actin monomers come together, they twist into a double-helical polymer called F-actin. This twisting structure gives actin its trademark strength and flexibility. Actin filaments have polarity: an A plus end where new actin monomers are added rapidly, and an A minus end where disassembly happens more readily. This creates a phenomenon called treadmilling, where actin is added on one end and removed from the other, giving the impression that the entire filament is moving forward. This is one of the core mechanisms that allows cells to crawl. But actin assembly is not random. It's regulated by a host of accessory proteins. Profilin promotes actin polymerization, coffulin accelerates disassembly, capping proteins stabilize the ends, ARP2/3 complexes create branching networks. Together, these proteins coordinate actin growth like an orchestra conductor, deciding when to speed up, slow down, build, or dismantle. Actin's most famous role is in cell movement, the ability of cells to crawl across surfaces, choose pathogens, or migrate during development. When a cell moves, it pushes its membrane forward using actin filaments in two key structures. Number 1, lamellipodia. Broad sheet-like extensions formed when actin branches rapidly under the membrane. These act like the cell's front foot, pulling it forward. Number 2 is filopodia. Thin spike-like protrusions that help the cell sense its environment. These act as antennae, detecting chemical signals and guiding movement. Actin polymerization at the front edge generates pushing force. Meanwhile, at the rear of the cell, actin interacts with myosin, forming contractile units that pull the cell body forward. This combination, pushing at the front, contracting at the back, is the fundamental engine between cell migration. We see this every day in wound healing, immune cells chasing bacteria, developing tissues during embryogenesis, and cancer metastasis. Wherever a cell needs to move, actin is leading the way. When a cell divides, actin takes on an even more critical responsibility. After the chromosomes separate during mitosis, the cells must physically split into two daughter cells. This final step is called cytokinesis, and it's powered by a structure known as a contractile ring. The contractile ring is made of actin filaments arranged in a tight band around the middle of the cell. Interwoven with these actin fibers are myosin motor proteins, which slide the filaments past each other, tightening the ring like a drawstring. As the ring contracts, it pinches the cell membrane inward until the cell is divided cleanly into two equal parts. This ensures that both daughter cells inherit the proper amount of cytoplasm and cellular machinery. Without actin, cell division would fail, chromosomes could separate, but the cell itself would never finish the job. Actin isn't just about motion, it's a major architectural component of cells. Actin filaments form a supportive scaffold underneath the plasma membrane, creating a cortex that helps the cell resist stretching and bending. Two classic examples show how essential actin is to cell structure. Microvilli in intestinal cells. Actin bundles from the core of microvilli, tiny projections that increase the surface area of the intestine for nutrient absorption. Without actin, microvilli would collapse like deflated balloons. Number 2, the shape of red blood cells. Red blood cells depend on actin-based network for flexibility. This allows them to squeeze through tiny capillaries without breaking. A defective actin network can cause the cells to become stiff or misshapen, leading to diseases like hereditary spherocytosis. So whether a cell needs stiffness or flexibility, elasticity or tension, actin provides the mechanical support required to maintain the proper shape. Actin isn't only important for normal cell function. When actin goes wrong, disease often follows. Mutation in actin or actin-regulating proteins can disrupt muscle contraction, leading to muscular dystrophies, immune cell movement, weakening the body's ability to fight infections, and wound healing, slowing tissue repair. Some bacteria and viruses exploit actin for their own survival. The bacterium Listeria, for example, hijacks actin and uses it to propel itself through cells like a rocket. In cancer, actin reorganization helps tumor cells migrate and invade new tissues. Understanding actin dynamics helps scientists design drugs that can slow or stop metastasis. Finally, actin-binding toxins like cytokinesis help researchers study actin's role in neurodegeneration, cell death, and tissue breakdown. Actin may be a simple protein, but its effects ripple through nearly every part of biology and medicine.